Nervous system and skeletal muscles. The effect of chloride ions on the nervous system and skeletal muscles. Gamma-aminobutyric acid, or GABA, is the primary inhibitory neurotransmitter in the mature brain. It reduces neuronal activity of target cells through its binding to GABA receptor, present on the cell surface. Nearly half of all synapses of the brain express some kind of GABA receptor and are thus responsive to GABA. There are two types of GABA receptors that we are focusing on, GABA A and GABA B. GABA A is a ligand-gated chloride channel. Upon transmitter binding, they open and allow chloride ions to flow into the neuron, making it more negative or hyperpolarized and thus less likely to generate action potentials. GABA B acts through a G protein to activate potassium channels, which allow positively charged potassium to flow out of the cell, again resulting in membrane hyperpolarization and the subsequent decrease in neuron responsiveness. GABA is believed to play a major role in controlling neuronal hyperactivity associated with fear, anxiety, and convulsions. GABA A receptor is composed of 5 protein subunits. This can be seen on the diagram along with the structure of both GABA A and GABA B. In addition to binding sites for GABA, it has allosteric binding sites for other substances known as GABA modulators. These are molecules that can increase or decrease the action of GABA, but have no effect in the absence of GABA. For example, benzodiazepines, a class of drugs used to treat anxiety, bind to GABA A receptor and facilitate its binding to GABA, thus potentiating GABA inhibitory effect. Other positive modulators include barbiturates, alcohol, propofol, among others. Examples of negative modulators are convulsants such as flumazenil. Flumazenil reverses the effects of benzodiazepines by competing with them at the same binding site on GABA A. The binding site of benzodiazepine seems to be between the alpha and gamma subunit to the GABA A receptor. Benzodiazepines do not bind to the GABA recognition site located at the junction of the alpha and beta subunit. Thereby, they do not induce opening of the ion channel as directly as GABA molecules. Instead, the binding of benzodiazepine to the receptor site increases the affinity of GABA to the GABA binding site. This leads to increase the frequency of the channel opening and in turn increases influence of chloride ions at the postsynaptic cell, which ultimately makes it difficult for any excitatory inputs to cause an action potential. Barbiturates bind to GABA A receptor at the alpha or beta subunit, which is different from the GABA binding site and the benzodiazepine binding site. This can be seen on the diagram. The benzodiazepine binding site and the GABA binding site are two different sites. On the following slide, you'll see a diagram where we include the ethanol binding site. Just like benzodiazepine, barbiturates enhance the effect of GABA at the GABA A receptor, although instead of increasing the ion channel opening frequency, they increase the ion channel open duration. In other words, barbiturates hold the ion channel open continuously for longer periods of time. In addition, barbiturates can reduce the activity of excitatory neurotransmitters such as glutamate and acetylcholine, and at higher dosages they can mimic GABA and directly activate GABA receptors, leading to greater levels of hyperpolarization. For these reasons, it is understood that the toxic doses of barbiturates are not much higher than the therapeutic doses. Alcohol inhibits responsiveness of neurons via its interaction with the GABA system. We can see the alcohol binding site on the following slide. Alcohol is known to potentiate GABA receptors, keeping the channels open for a longer time and thus exaggerating this inhibitory effect. GABA receptors are also the target of certain anesthetic drugs. This explains the sedative effect of alcohol. It is now obvious that the combination of benzodiazepines with alcohol or barbiturates will amplify the hyperpolarizing effect due to prolonging the chloride ion channel opening extensively. This could lead to various health implications in future. As mentioned previously, Movement of chloride ions is based on action potentials. These ions move through a ligand-gated channel specifically for chloride. This aids in repolarizing the cell as it allows greater movement of chloride ions out of the cell after hyperpolarization. One can now only assume what may happen to the cells if this channel experiences any implications. Some pathological conditions associated with chloride ion channel impairments or mutations are known as channelopathies. Myotonia congenita is the most common type of skeletal muscle channelopathy and is caused by mutations in the skeletal muscle voltage-gated chloride channel gene. In myotonia congenita, when we reach the action potential threshold, we have the normal opening of sodium-gated channels depolarizing the cell with the positive charges of the sodium ions. Although the deformed gene prevents the chloride ion channel from being formed completely, this leaves only the potassium-gated channel to repolarize the cell or enter the state of rest or resting potential. This condition keeps the cell depolarized for a prolonged period of time, keeping the muscle of the individual for a longer period of time, leading to freezing of the muscle with the inability to release or contract.